Our next researcher is Dr. Jacques Tremblay, who is professor in the Department of Molecular Medicine at Laval University. He has worked on cell and gene therapies for hereditary disease, in particular Duchenne Muscular Dystrophy, since 1987. He has worked mainly on the transplantation of myoblasts derived from a healthy donor as treatment for Duchenne. This evening, he'll give us an update on his current research on in vivo corrections by CRISPR prime editing of mutations responsible for Duchenne muscular dystrophy. Over to you, Dr. Tremblay. Oh, you're on mute.
On mute.
There you go.
Can you hear me?
We can, thank you.
My current research project is called in vivo correction by CRISPR prime editing technology of point mutation responsible for Duchenne muscular dystrophy. In fact, the human genome contains 3.2 billion nucleotides, and there are only 4 types of nucleotides. They are designed by ATCG. And the modification of only one nucleotide may sometimes be responsible for an hereditary disease. In fact, in the case of Duchenne muscular dystrophy, 30% of the patients have just a mutation of one or just a few nucleotides. And so all the rest of the gene is correct. Can we correct these mutations? 70% of the patient with muscular dystrophy, Duchenne muscular dystrophy, they have a deletion of one or several exons, which is changing the reading frame. So my research project has 2 aims. One is to correct the mutated nucleotide, there might be one or just a few. And second, to restore the normal reading frame of the dystrophin gene in the patient having exon deletion. And this could be done by inserting or removing just 1 or 2 nucleotides. So all of you know, in Duchenne muscular dystrophy, due to the mutation in the dystrophin gene and the DMD gene, there is no dystrophin underneath the membrane. The prime editing technology is a fantastic technology that was published for the first time in Nature in 2019. It's using an enzyme derived from the CRISPR-Cas9 technology. It's called a Cas9 nickase, and it's an enzyme able to cut only one strand of DNA. And this enzyme is fused with a reverse transcriptase. This is an enzyme that is able to make DNA from a template of RNA. And this technology also uses an engineered prime editing guide RNA. And this is the important molecule. It has, in fact, a sequence of 20 nucleotides that will hybridize with a sequence in the human genome. So although the human genome contains 3.2 billion nucleotides, a sequence of 20 nucleotides is specific and will exist at only 1 or perhaps 2 sites. So this is one way of targeting a very precise sequence, in our case, within the DMD gene. And then there is a constant part of that PEG RNA. And then there is an additional sequence. One is called a primer binding site. It's a sequence of nucleotide that will attach to the strand of DNA that has been released by the nickase. And this is followed by the reverse transcriptase template. Again, it's a sequence of nucleotide. But this is an important sequence because when you change some of the nucleotide in that sequence, you can also change what will be the new DNA strand that will be incorporated. So if you put a T, for example, in that reverse transcriptase template, this will produce an A in the resulting DNA. So with this reverse transcriptase template, this is the trick to be able to correct any selected nucleotide. So here we see the complex being formed. We have the PEG RNA making a complex with the Cas9 nickase and the reverse transcriptase. And this will then attach to the DNA. It will recognize a very specific sequence in the DNA. In our case, we're targeting a sequence within the DMD gene. And this will permit the synthesis. That you see the primer binding site here is attaching to the strand of DNA that has been released by the Cas9 nickase. And this reverse transcriptase template is the template to produce a new DNA molecule that will be and this DNA molecule will contain the modified nucleotide to correct a point mutation. So following this treatment, we have a strand of DNA where only one nucleotide or two, depending what we want to do, has been modified. So it's a very, very precise technique that can be used for Duchenne muscular dystrophy, but can also be used for several other hereditary diseases which are due to the mutation of only one nucleotide. So in the case of the project on DMD, we are looking at 3 mouse models of Duchenne muscular dystrophy. And these 3 mouse models called MDX, MDX4CV, and MDX5CVs are all due to the modification of only one nucleotide, which in this figure has been put in red. So using this approach, we've been able to introduce with prime editing technology, we are able to either insert a mutation in the dystrophin gene, or we can correct a mutation in the dystrophin gene. So we've done both type of modification in my lab. In this experiment, for example, we were able to insert a very precise point mutation in 20% of the cells of a MDX4CV mouse. So for each of the mutation that we want to target with the prime editing technology, we have to produce up to 9 different PEG RNA, where what's changing from one PEG RNA to another is the number of nucleotide in the reverse transcriptase template and the number of nucleotide in the primer binding site. And the change of these number of nucleotide has a profound effect on the capacity to correct or not the targeted mutation. So it's very surprising. And in fact, in some recent article, they are even using 100 different PEG RNA for each of the mutation that they want to target. So we have already produced these ePEG RNA to correct mutation in the MDX mice and in the MDX4CV mice. And for the future, the main problem of this fantastic technology is the delivery. As they always say in gene therapy, there is one problem: delivery, delivery, delivery. And so the problem is that the component of the prime editing technology is too big to be delivered by only a single AAV, adeno-associated virus. So we need to use 2 AAVs, and this will reduce the efficacy of the treatment and will eventually increase the price. So we are looking at other delivery methods. One of them is the lipid nanoparticle. It's the type of particle that were used for the COVID vaccine made by Moderna. And so these lipid nanoparticles are very good at delivering messenger RNA. And so in collaboration with the McGill University researcher, we're now trying to make lipid nanoparticle that will target preferentially the muscle to deliver the prime editing component so that we can correct point mutation within the muscle. We are also using extracellular vesicle, and extracellular vesicle are very small vesicles that are secreted by all type of cells. And in my lab, we've been recently able to modify the cells producing the extracellular vesicle so that they contain the prime editing protein. And so this is one way where we'll be able to package the Cas9 nickase fused with the reverse transcriptase and the ePEG RNA. All of that will be packaged into microvesicle or to exosome, as mentioned previously. And this is another method, therefore, on which we are working. And finally, we are also working on a method called virus-like particle, VLPs. And these are modified retrovirus that instead of delivering DNA, can deliver protein. And again, these virus-like particle, we've been able to introduce the Cas9 nickase fused with the reverse transcriptase into these virus-like particle. So I think that if we can develop these delivery method to be very effective, this will be fantastic. This will permit eventually, we hope, not only to cure a patient with the point mutation in the DMD gene, but the same technology could eventually be applied to other muscular disease and even to disease which are not muscular disease. Thank you.
Thank you so much for your update. I really appreciate it.